given our swabs the weekend to grow on our Petri plates, it's now time to look at our bacteria colonies and analyze our data. A bacteria colony is a group of bacteria cells that came from one single bacteria cell. That bacteria cell was given enough time to grow, in this case about 36 hours, and now you can see it without the aid of a microscope. That single little microscopic bacteria cell is now a macroscopic group of cells that we can see. We're going to look for a couple features on the bacteria cell. One is the shape. Is the colony circular, irregular shape, filamentous, or a rhizoid? That is how we'll categorize the shapes of the bacteria colonies when we observe them. We're also going to look at the edges of the bacteria colonies, which is called the margin. If it's a nice smooth edge, that's called entire. If it's kind of a varying edge, that's called undulate. A filamentous type of bacteria colony will produce filiform edges. You can have wavy, kind of curled edges. And you can have lobulate edges that really extend out from that central bacteria colony. We are also going to observe the color of the bacteria colonies. So we're going to look at the form, the margin, and the color of the bacteria colonies as we count them. So here's one of our Petri plates that sat out over the weekend, allowing bacteria to grow. You can see a lot of colonies are circular with entire edges. But we do have some filamentous bacteria colonies with filiform edges, and there are even some irregular colonies with undulate edges. Now, you guys are limited to looking at a picture, so I'm going to give you quite a bit of help identifying the colonies, what form they have, and what margins they have. You should be able to look at the pictures and get the colors of the bacteria colonies. This is the third page of your Bacteria Project Lab. You will have to insert a picture of your Petri dish here. Then you will have to analyze that picture and identify the different forms of the bacteria colonies in quadrant 1, 2, and 3, the margins of the bacteria colonies in quadrant 1, 2, and 3, and the different colors of the bacteria colonies in quadrant 1, 2, and 3. For the last data table, you want to organize the surfaces that were swabbed for each quadrant, how many bacteria colonies you found in each quadrant, and the different types or colors of bacteria colonies you found in each quadrant. So to help you with all of this, I created a Google Slides presentation. I have 10 slides here, one for each of the Petri plates we created. So all you need to do is use this Google Slides presentation to help you fill out the data on page 3 of the lab. The first thing you'll want to do is just select that picture of the Petri plate, copy it, and then paste it into your document. Then you're supposed to identify the form, margin, and colors of the bacteria colonies in each quadrant. I've helped you quite a bit here. I've given you the form of the bacteria colonies in the quadrant and the margins of those bacteria colonies. So the information I have written here in green is the form and margin of the bacteria colonies in each quadrant. You will have to look at the pictures and find the different colors. The colors range from white to orange to tan to red. That is all the color variations you're going to need. For the last data table, you need to write down the locations that were swabbed, and then I either told you how many colonies were in that quadrant, or I circled the bacteria colonies in that quadrant, and you have to count the circles. You also have to look at the picture, see how many different colored colonies that you see for that third data table. All the information you need to complete the third page of the lab should be on the Google slide for your Petri plate. After collecting all your data on page 3 of the lab, you need to finish the lab by completing the analysis questions. For the first analysis question, you must address the predictions that you made. So which location did you think would have the most growth, the medium amount of growth, and the least growth? So when you look at the first analysis question, you have to write down how many bacteria colonies were in each quadrant, 1, 2, 3, and 4, and then if that supports your hypothesis. So if you thought the location swabbed in quadrant 1 would have the most bacteria, did that support your hypothesis, or was your hypothesis incorrect? And then you must explain your answer. I am expecting specific answers with complete sentences, capitalization, and punctuation. Spend some time on this first question writing specific responses. The remaining questions should just make you think a little bit about the lab, like what was the purpose of our control in quadrant 4? What are some reasons why there might have been some bacteria growth in quadrant 4? Do you think if we swabbed for bacteria at your house that the results would have been any different? Explain yourself. Then make sure to go and look at the Google Slides presentation at all 10 of the slides. Some of them are really interesting. All I want you to do is find one thing that surprised you when you looked at these Petri plates and explain it to me. Make sure you are writing in complete sentences. A complete sentence starts with a capital letter and it ends with some type of punctuation. I expect high quality work.